You are welcome to Bio 201, the Plant Physiology Unit 3, Session 6, Demonstration and Effects of Water Movement in Plants. And by the close of this session, students should be able to show osmosis in living plant cells and determine the mean solute potential of the cell sap using the method of incipient plasmolysis. So we are good to go. Demonstration of osmosis, the concept. We have noted that osmosis is all about movement of water molecules, which is made possible by the inherent kinetic energy possessed by the water molecules. This means that water molecules have the tendency to move about in a system, and that when two systems are separated by a partially permeable membrane, the rate of movement of water molecules in either direction will depend on the kinetic energy content of the water molecules on either side of the partially permeable membrane. So the side of the partially permeable membrane that has the higher concentration of water molecules will have a higher kinetic energy, otherwise called water potential, which will cause a net movement of water molecules into the side with lower concentration of water molecules across the partially permeable membrane. The typical plant cell has cytoplasm and vacuolar cell made up of dissolved salts at varying degrees of concentration. That makes it susceptible to fluctuations of influx or loss of water. When conditions are favorable, like being surrounded with an environment with high water potential, there will be a net inflow of water to cause the cell surface membrane to stretch off firmly against the cell wall. Where the reverse is true, the environment being unfavorable, the plant cell will lose water and cause the cell surface membrane to move away from the cell wall. So, do water molecules move along a concentration or potential gradient in living plant cells? Does the influx of water into and out of plant cells have effect on plant cells? To investigate osmosis in living plant cells, this activity is taken using the skin of the onion bulb or Rio discola leaf or Rheospathacea. The activity, you have materials, leaves of Rio discola or onion bulb. You have microscope, slides and cover slip, scalpel and forceps, distilled water, 1 molar sucrose solution, pipettes, filter paper, and glass straw. The method is that Rio discola leaves must be obtained fresh from the garden. Carefully wash off dirt under running water or a trough half filled with water. Use a scalpel to make a small indentation on the outer surface of a Rheospathacea leaf. Use the forceps to carefully pick and pull off the skin, the epidermis, from the edge of the indentation. On the other hand, you could fold the leaf to break it, and then with the fingernails or forceps, peel off the skin, epidermis, from the broken edge. Quickly transfer the epidermal strip to a slide. Add 2 or 3 drops of distilled water. Gently drop a cover slip onto the water-covered epidermal strip. Mount and examine the cells under the microscope, first under low power, then high power. Mount another epidermal, this time in sucrose solution. Then you leave for about 5 minutes, then observe under the microscope. Irrigate the amount in 1 molar sucrose with distilled water to wash away the sucrose solution. Use filter paper to absorb excess liquid until you are sure all the sucrose has been washed off or replaced under the cover slip. Then you observe with low power. Observations. What did you observe of the plant cells under low power magnification? Make a few low power drawings of the Rio discola epidermal cells and label appropriately. What differences are observed between the cells mounted in the 1 molar sucrose and the distilled water? Make a label drawing of the Rio discola epidermal cells to support your observations recorded in 1 and 2. Then What differences or similarities can you make between the plant cells after irrigation with the distilled water and that of the distilled water at the start of the investigation, and the cells in the 1 molar sucrose mount? Make a table to state your observation. Well, in conclusion, what conclusions can be drawn from the state of the plant cells under varying surrounding conditions in response to changes in the water potential around it? Determination of the mean solute potential of plant cells using the incipient plasmolysis. The concept incipient plasmolysis describes the state of the plant cell which, as a result of it having a higher water potential than its surrounding, lose water till the cell surface membrane just begins to shrink and to pull away from the cell wall. When conditions are changed, the process of plasmolysis is usually reversible without permanent damage to the cell. How can this process of plasmolysis be determined for in a plant cell? So activity 2, using incipient plasmolysis to determine the mean solute potential of living plant cells. So the materials we have: real discoidal leaves, petri dish, scalpel or razor blades. Prepare different solutions of in 2 milliliter flat bottom flasks. You have distilled water, you have fine forceps, slides and cover slips, microscope, you have a graph paper, then you have guide pins. So the method is that you arrange in a row labeled prepared solutions of sucrose in flat bottom flask. The sucrose concentration should be 0.4 molar, 0.45 molar, 0.5 molar, 0.55 molar, 0.6 molar, and 0.7 molar. Line up 6 petri dishes in front of each solution. Pour some of each solution into the petri dishes. Fold over backwards to break the real discoidal leaf on the outer surface, or use a scalpel to make an indentation on the outer surface of the real discoidal leaf. Then use the forceps to peel strips of epidermis from the inner surface from the broken edge or the edge of the indentation. Cut each strip you peel off into squares of 5 milliliters by 5 milliliters. Place 5 of cut epidermal squares into each petri dish with the different concentrations of sucrose solutions. Place 5 of cut epidermal squares also in a petri dish with distilled water. Use a guide pin or the fine forceps to pick a square strip and mount in the same solution on a slide and drop a cover slip on it. View under low and high power of the microscope, starting with epidermal strips in the sucrose solution with the highest concentration, and move on down to those in the distilled water. Count the number of cells in each strip recorded. Count the number of plasmolyzed cells in each strip. The colored sap enables easy detection of plasmolysis. Here, consider any cell in which there is any sign of the protoplast pulling away from the cell as a plasmolyzed cell. Make a table to record your data for each sucrose concentration. Then you find the mean of plasmolyzed cell in each different concentration of sucrose solutions. Use your data of mean percentage of plasmolyzed cells to plot a graph against the different concentrations of sucrose solutions. Determine the sucrose concentration that causes incipient plasmolysis in the real discoidal leaves. In review, you note that incipient plasmolysis is said to have been reached when 50% of the cells have plasmolyzed.